Okay, so this is a tour of the wiring of the brain.
That they take the what they carry la nanga lepag nala ya chi dretu kanado chi say take and it you telaya and you attacker yes.
And a lot of advancements in neuroscience have emerged from our ability to have these technological breakthroughs to probe the brain.
That once a Henry Shimji la ya chi Yapuchungu Yobati Sukares and Asuichi Lepi Nanlaya could Jetu Kadadochi drew those at Take Kanlaya, Taxetia Getachi Tlet and Triri or Trichi or Triri de Yerji Timbalatanichongares.
And my goal in this next section is to introduce to you some techniques for studying cognitive neuroscience, which is the neuroscientific investigation of mind.
Tandagi Lomton de Nalaya, to Loptrichia, Midi Karin, Sena de Capsa, to once a Henry Sigurd that the Dig Corlaya, Motorchigins, once a Henry Dig Telaya, so the answer means once a Henry Segura, means once a Henry Sena, so the Garsagorasena, Tanda once a Henry did Peshutan data, and then I took a lepin on the Laya, Leka Kanaruchi do search for a take or a Tassetia, take or Motorchigins.
This will be a very kind of general overview, but we'll be focusing on some of these techniques for a lot of the sustainability phase, and want to provide opportunities to you to get more involved in working with these techniques if it interests you.
And it's Trichi Chetapa and Koga Leka Chetan Chetapa in the Dola, Nathan Canado in Badechula, Lantra, Carsueta Koto Laya, and it and then a Sulerum Diganalaya, a younger is.
So, for some time now, there's been a rich conversation occurring between cognitive neuroscientists such as Richie Davidson, that you see here, and His Holiness the Dalai Lama, Mingyur Rinpoche, right, Mingyur Rinpoche here, and Matthieu Ricard.
Now that Tandangasu means once at Saint Sigurd, Talepi, Ganlaya, Tashichia, Chigoro, Gontan, Jerry, Tashichia, Digitalaya, Chitaxepi, Tachi Chichi, Otsukorio, Restata, the big Ganlaya, Tashichini, so the Richard Davison, and it take Anetanda. Changurumbuchi Cherwa, and it and the Minjurumbuchi, and the material cut Kosu, Parola, Tanda, Chigansu, Lepala, Tasechia, and Tilkolaya, and the Kamuti, Ayansi and Samampuchon.
And the idea here is that these two traditions of academic neuroscience and Buddhist scholarship could really come together to work together to investigate the mind.
That and the Dignan is so cheap, Karitangoris, so once such like a taxi, taxi data, and a Jinambigi, Jikebatan, Jinyalam Bakuso, and a Nyamton Yamrichuala Tene, Negaso Gilepata once Sagitelaya, and Taxi Chitpati Dignan Tangioris.
I am personally very excited about this conversation. I did my graduate work with Richie Davidson. And so for a long time, I've been interested in the conversation between Buddhism and neuroscience.
That Nang any once out Senipag Parugi, Limodi Korlaya, Ngara in Banat, Tonatria, Tantela, Shuchi, Gatuchin, Sangara, Womati Karsata, Tularam Jambas, Tularam Jambatanta, Tulaki, Tomitin, Chivi Capsi Baina, some to Jiatian and the Dukolaya, Colobjan Chiwatan, and a Colobjan Chisati again, than the Richard Davis Singola, those Lojan Chishavachins.
And I'm very excited to work with all of you. And if any of you are interested in more in-depth training in neuroscience, we will be coming to Drepung to help you train in neuroscience. And some of you may have the opportunity to come to my laboratory in the United States and work with me and Ken Paller to train in neuroscientific methods.
once Tsering de Kalata le patang semcote la ya Shimji Chiwate Chitutanyam Chitwate Hachingatua Shu Pegtus that and then by na Chini Natso Pa Depola Yonchi Chitula and the Dick Pelaya let you think Sapo on the Shimji Chia and you chit Sungotrochia get Netantia Yongrella and the Nanshingi ni Chetsuna and Mikashi Kashi and it's Sunkore those Arila paging and so get Tashikala Yonchi de and again can power down and so get Tashigi Kalaya Tigola Yanchi Tashi things of good Rebecca Cocatin Ragris.
Now, we've learned a lot about self-report measures for studying human experience, mind, and emotion.
Tanda Patangasu Karikuto Chonores Tambala Natsuge Kasugre, sentence, Susunyawata, Tidado, Kolaya, Shinjichi, we have the self-reports, Sususu, Moneman, Kutushi, Sususu, Kutushi, Lenti Kolaya, Ningatsu dus Chitayores.
Self-report has many strengths. Like all techniques, it has its limitations. What do you think might be a strength and or a limitation of self-report techniques to study human emotion and mind?
아니 지금 유치 찌던 총와라 양아수 self report 쓰고 수수수수 군만큼 미담보 간소 담보고는 레지시샤 주게 지금 레지치 거와 어때라야 아니 그 델라야 그 지가스 그랬다 신차다 김팽이 요리스트 제츠타바인 바나 수수군이 다 그르긴 레지시샤 주게 년투스 거라다 게줄게 년투 델라야 아니 지금 긴센다 양고 스튜언스 거 리미테이션 시스템 발라 체크가 라진 가리가리 요사리스 델라
Anyone can just kind of provide a thought.
Hello.
Hello. Tashidale.
Yeah, Tashidale. And I think the benefit of self-report is that if we research on the emotions, that is a subjective. So it is the first person that is the most reliable one. That is the benefits of self-report tool. And the challenge is that one, if you self-report, they might be telling lies if you're not honest. So this is the limits of self-report.
Okay, na sorona.
Self-report kalam.
Graduate nyanto do labna.
Karso. Gerjoge nyanto da keben karerisa ba ina penanga su senyom da de so gola ya da so michile ina so so gi nywa de so so gi da yakshe ha ko ro jesan digi ni su de tambo dang sha ba ina ni su di mi thoma de ya raya jora da yakshe de chago re ta de jancha gare yo se wa ina mi digi zuma she si re ya so so gi ya ngo cha ya da ya dindi wang gi ya tak da ma she gi ne ga yo ra di xian xia de tong gu de to xian
Thank you.
So I think that's a phenomenal response for a few reasons. First, I loved the statement highlighting the importance of self-report, because self-report is our own subjective experience, and that is so important in the investigation of mind and emotion.
Yapu ching dishas right todo chison son testambalaya kechimriseti ta junsinchi kare testambalaya chingatsuge ta gerjoge nyentu tion stambalaya tambo de sosu gu nyawate sosu gu jopa imbana kota ya shu yabu jetu baina ani tela ya tus tela tende te sosu gu nyawa yansa tela tigi gempenti yores.
Two limitations. So one limitation that was mentioned is that people may not be telling the truth, and that is a limitation of self-report.
Da shendachi karayore system bala ya degi chu ko chende karayore sana chichina yang ko deshi susu deshi jagu yorda imba na ya mich degi deshi jabi kapsa demba shegi yona mena sayati yang ko geta chi cheka karanzo chunchi res.
But there's two other limitations of self-report that I want to mention.
Dada de gejo yanto de la ya ko ge ta cheka karanzo jenda nis yore teo deshi janji to sons.
The first is that we may not have access to the kind of unconscious processes that often can generate emotion.
Da shenda bana chikari yoresena ngasu ge chita samshin to mind basegoro da samshin to mind bi gane ane ji samluna chong chong atela tene de tanga su samyon chi string yo page ni tan tene da uchila yang ngasu susu ga hama ko ge gane chong achoro te tanga su te hama ko ge ne tan tene chong gres.
And the second is that we may not have access to the mechanics, the kind of fundamental principles that generate our emotions and thoughts.
Ya shenda chi kare mangyo tenda yung gre system bala ngasu ge chiga samyon ta samlu ti tinya ge jista ge chi sanjin da kari chi ba ti ge tentang ge chiga dojam da kanda da chi ba ti hama ko tina yung gres.
And so combining self-report with cognitive neuroscience methods is a potentially powerful way to take the best of both techniques to really examine the mind in a more comprehensive way from a scientific perspective.
Tanda teens tambala ya ngachu ge gerjo ge nyento te che te gangla ya ani minzen wansa chenri ki tablan ji orwa ten chan nyam te jete tang ba imba na nyam te nyam ri gang ne jiba imba na ani ngachu ge ji ji ge lepa ge drotan ta ngachu wansa chenri ko ngu drotan te la ya ani chi ge phajo chejok pe ji kar sabure ta ji khasib ngu ni ani chi ngachu samgi sibura ta lepa ge nang drotan te ngunsu to bu gang ne jejo jaga la ya nyam ri gang ne pengores.
And I will say that I posted these slides on the Google worksheet. So if people want to follow along, they can.
That de tanda de la ya de chi do slide chi do de tanda chi chu ge zinjagi na la chi chu Google ge chi do de na tali ani question yus ni chi chu de la tanda de tanda dires.
So in our work, we use both self-report and cognitive neuroscience methods in a complementary way.
Da nga chu ge le ka gi na la ya tanda gerjo ge nyen to te ta ane ngin zon wa sa ser ge tabla ni cha na pansun la ya chi roram ji ngu ga ne sura pansun ji tunjo ji ngu ga ne nyam te nyam re ga ne pe chi tang yes.
Now, neuroscience and the field of neuroscience involves multiple levels of analysis.
Da wanza chenri ki nyam shi ge na la ya na chu ge ji Egypt ji ya ge ji ramba ma so na ramba mampu chi dus na chu ge yes.
You have some people who are really focusing on biological or biochemical processes kind of at the tissue level of the brain.
Da kashi chika kange ga dona chika reses tamba nga chu puntu ku rambala yope gi ani chi chingwe zejur gi ani chi jurum ta chi joran do ya chorwa ti ga la to na chi chi yores.
Then you have people like Ken and myself who tend to focus more on systems level human neuroscience.
Da again ken la ta ngaran ta so ba na to na chi sa de ani chi malak ki ramba ga la ya ani karsubure ta ti kapsa once I malak ramba de ga la ya ko chapter current do se ti la tak se che la mus res.
And then you have psychologists and others who are interested in kind of really intersecting neuroscience with psychological studies and even social experiences.
Da shemba chi kare ores tamba nga su samkhan ru pa wa ku su in ba na ya wangza gi jingro tang de ba kan shi la ya mi gi chi ta nga su samkhan tang da wa yu chu ne chi chu tang da wa yu chu wa ti ga la ya ku su ge ta shi ji chi res.
At the molecular level, there's been some wonderful advancements in looking at the structure of brain tissue, brain cells, and brain function.
Da dide ge ramba ge takshe ge tela ta ba ba na ku su la ya ji shu chu du yo resta de kap se ba na ji nga su ge la pa ge chapon ta ani shele ta ko ge phung tu ge ri te la ya dus takshe ji thi yo res.
And this involves both light and electron microscope or microscopy techniques that can probe deep into the tissue of the brain.
Da de kapsa zo de nga su kar pe chi tang gore se stambala ya thatong chi she ge trug de pe chi tang gore sta ti pe chi tang de ta de na la ya nga su pe chi tang ya ge thatong chi she pe chi tang gore ti na shin ge log de 차통 치슈도 배치던 거. 티 배치 땅에다 가르치고 그랬으나 다 왕자기 차시베데 참을라 디데던 거기 내단텔라야 다시 그랬어.
And I just want to give you a taste of one of these techniques that is frequently used called light sheet microscopy.
다 단다디 값을 그속에 딜라야 아니 light sheet microscopy 쓰고도 왔다가 주 여기다 왜 당와라 되는데 아니 그것을 차비니스라 다지고와 어디레스티데
which allows us to do a deep dive into the cellular structure of brain cells called neurons. Here you can see a neuron. These are all the synaptic connections that are connecting those neurons. And these advancements have really allowed us to understand the cellular structure of the mind.
디나나 단다치수 토바데가레스나 가수가레 피기난 그 타폰스거다 왕사 타폰 티다다 왕사 타폰 판승길라 네 지금 투접지비게 네탄 카나도요 파틴도 스탱기오르스탄 데임바나 가수게 지금 다 샘스나 두그레 레파게 다치 타폰가 름발아야 구축 네탄 카나도 지금 요 파테 터스지알 팽기오르스.
People study also different species to understand the mind. Here is a printout of a human brain. This is a macaque monkey brain, and this is a mouse brain.
다 같이 인바나 가수게 지금 샘티 딱시지에 제때요 레파 지가스 니르 만도게 레파 터스지고리스 지소데 디 가수 미게 레파지 챈다디 비우지 챈다디 가수 지지 레파리스.
My goal for today is to just give you a very kind of brief introduction to techniques for studying the human brain.
대랑게 무예데 가린스나 미게 레팔라야 딱세지에 값서 아니 지금 트르마소나 트르당 답런까지 배치치던 거에 모르지인스.
And this is often called the field of cognitive neuroscience.
근데 지당에 말들랑아 좀 인증 원서 저렇게 가서 그랬다 그게 린슨스 그랬어.
And again, my hope is that some of these techniques might interest you or excite you, and that some of you who want to learn more about these techniques could train with us and become collaborators on research projects of the confluence between science or between neuroscience and Buddhism.
지치나양 단다가 속에 지금 답은 트르긴다로 비치던 담발라 가시양 되게 트르게라야 그래서 그래. 지금 도양을 해봤다 아니 그 야포 통화되는 지대대 아니 그 디게 콜라야 미트도 양고 하장 거서 띵사 보시니도 어떤데도 요그래더도 여바이나 양 제마 나다가 소다 남바다 네 왕사 전립바가 바로라야 지금 냠제기 냠레지야 저와 어디다가라야 가서 냠리지에 그때야 용그랬어.
So in these next few slides, I just want to give you an introduction to the specific techniques that I'll be discussing today. The first is a technique called electroencephalography, which we'll call or refer to as EEG.
And
EEG is a very effective way of studying brain potentials on the scalp of the head. And it's a very effective method for studying how active or deactive a person's brain is at any particular point in time. And other techniques of EEG allow us to also probe certain psychological processes or neural processes in deeper ways. Another very common technique in the field of cognitive neuroscience is magnetic resonance imaging, which we'll refer to as MRI.
there is.
Now, structural MRI allows us to take a very detailed picture, almost like a map of the human brain.
Structural
and provide a very deep window into the structure of human brain tissue. One of the limitations of structural MRI, though, is that it's just a picture, a static picture of brain tissue. So another technique that we can use with MRI is called functional MRI. And functional MRI allows us to actually examine brain function as opposed to just studying the kind of static tissue. And the last technique that we may or may not have time to get to is what's called diffusion imaging. And all I'll say about this is that this technique allows us to study the wiring of the brain, that is the connections in the brain. Now, one of the big, well, let me start over. One point that's important to make is that each of these techniques has their strengths and weaknesses. No single technique is perfect. And a few different domains of strengths and weaknesses are as follows. The first is what's referred to as spatial resolution. In spatial resolution is the precision with which you can identify or localize where in the brain the signal you're measuring is coming from. So as an example, you may have heard of the term the amygdala, which is a brain region that's involved in fear. A technique that has good spatial resolution would allow you to actually measure the activity in the amygdala during experiences of fear or other types of emotion. Another important domain is what's called temporal resolution.
And this
is the precision with which you can measure the timing of a signal that's coming from the brain. The brain, or I'm sorry, a neuron in the brain typically fires about 400 to 500 times per second when it's really active. Which is super fast. So temporal resolution is the ability, is the extent to which you can measure the really fast timing of the brain. Now, the final domain that's important to consider is the invasiveness. That is, how invasive is it to study?
Invasiveness
And I will say that all of the techniques that we'll be discussing today are very non-invasive. They're designed for human studies that are not invasive. Invasive here means that they don't involve looking at actual tissue or having to go into a person's skull or do anything that kind of, might hurt the participant. And so for each of the techniques I'll be describing today, I'll discuss their pros and cons on spatial resolution, temporal resolution, and invasiveness, although none of them are invasive. So just spatial and temporal resolution. And again, in my laboratory, we use all of these techniques because each has their own strengths and weaknesses, and we like to combine these techniques into what is referred to as multimodal neuroscience. Now, this is a very complex figure. I don't want you to get lost kind of in the details here. I'll just make a broad point. And the point is that you can really rank each of these neuroscience methods on spatial and temporal resolution. And that's just the main point I want you to take away from this particular figure. And one technique might have strong spatial resolution, but not good temporal resolution, or vice versa. So the first technique that I want to introduce you to are some of the techniques that we'll be focusing on more so in this particular training program.
다 단다데 current decision to some system balaya ani tigi tablen chiche tigna ne hapart tanda ngasu tanda chiga 심지 chigo detan ro chi tamen chinguro chigins.
And these are techniques for measuring the electrical output that the brain, or these are techniques for measuring the electrical signal from the brain.
다 단다게 대가래라 나 ngasu ge lepe na la yo pe logda chigo logda ti chenje ge tablen teres.
So these techniques all measure essentially electrical output that the brain generates through the firing of neurons.
다 단다게 대가래 지그레스나 ngasu lepa ge na la ya chi log tang devi ge dati cholola lue chigo ote ani chenje ge ore ti loti kanat chungo re system bala yongwa te ya chi wangsa ge datin ti pha tangwala tin chungo res.
So the first technique that we'll discuss is electroencephalography, big word, we'll just call it EEG.
다 당보데 럭스 let's go longe set is to the team by not to not touch it doing but under the caps in chicks do by any EEG.
Now, EEG measures, as I mentioned, the electrical signals coming from the brain, and electrical signals are sometimes referred to as electrical potentials.
다 단다 되게 let's go longe 되게 right EEG 되게 가리치그레스나 레판에 좀 베게 록다티 아니 고 체제기요레 아니 상상은 가수게 지가 록다 데라야 록다 데라야 가수 다록 니체세 포런시세 지거와 어디양 라그레스.
And I'll show you some examples of EEG equipment, and I'll tell you that we are currently shipping 12 EEG systems to Drepong for you to use.
단단히 디 let's go logic 답은 티페주 당하기 추치된다 만도 바카시 중고 추치기 인스와 니 데이터 쇼라야 단다 아니 고 양고 단다 드레로 탑 디우 근데 피주 당 퇴하기 EEG 된다 존식 단다 네 단다 데볼로스링 기체니 딘살라 두스 쿠르데타 아니 치추 피주 당 퇴하기 제때 두스 쿠리스.
And each of the monasteries and nunneries will be able to take one of them when they go back, like to Sarah May or Sarah J. So we'll each one should be able to have, or maybe they'll all stay with the song. I should retract that statement. We're going to work out the details, but you all have the chance to work with them. Never mind. I'll retract that statement. So let's go back to my description. An EEG cap is something, or an EEG system is something that involves electrodes on the scalp.
다 단다게 데라야 EEG mala sekdwa let's go longe mala de chigkalchigresena gogan gopa kanlaya ani ko logne youtube chichigula konya chigres.
And it measures brain electrical signals that are emitted through the skull and picked up on the scalp.
다 되게 kalchigresena ngasuge lepege nangne chongwege logda chigorwa logda de je ani lepige lochug logda se doda te lepige ta papa ganlaya ko logneti kombala tente tati juguyores.
And the simplest way that I can think of to describe EEG is basically a metric of how active or deactive the brain is.
다 단다 de kasanga su let's longe EEG sana ko tab desho gone deshe jabba imbaina lepate ji karasugre ta trompo rendo mundu se chejagi netan chires.
So if you were to think of a car, this is not an actual measure of EEG, but if you were to think of a car, in a car you have revolutions per minute, which tells you how fast the engine is firing.
다 단다 de pisha baina de muda chila ta baina ro muda langkhor chila ta baina langkhor te na ya ani chi revolution per second system bala ekra de na tele ko kora katsu chi jag do se korta de dodo she baina ga su muda na la ko chuchi ama chi ro chuchi ama te ge ne ko nipa te shu chilo chiteg do se te changores.
And this is kind of the similar for the brain, as you can see here a map of the distribution of electrical activity on the scalp, and this reflects essentially how active or deactive the brain is on those portions of the scalp.
That and that dig data chats on going as with dig on like Suzuka dig on like a lepig gun like a lookda monglu take do such doa or did ge tit on Tumbig and Nico party race or Sapra Kosha race that dig Gani Kukari Songo race Stambalaya lepig chashi kangi kangi salon chisha kangi salon chimadu salon cheese machi said it's head on again near which race.
So EEG can be, you know, fairly complicated, but to just make it very simple, I just want you to be thinking of it as a measure of how active or deactive the brain is.
That and the EEG, let's all get in Banachitang in Bana, not as how in the doji English race in Bana, Tanda Chesu, Samluna Niguati, let's go longevity Latin de ni lepate, Miss Helon, Katsuchi Chidu Sura, Salon Chedo, Salon Chimado, say, take it 10 to Korochis.
And this is actually a picture of an EEG cap.
That other day, like personal, Tandanga Sugelesi, Tege, YouTube,
So the blue part here is the cap. It's like a swim cap. It's like just an actual cap. It's a cloth cap or mesh cap or something like that.
That they like checking on which to go, but they children as a shamu to the door. What you get Tambala Shamu and the door with Shamuchis.
And then each one of these little white things is a specific electrode that measures the electrical activity emitted from the brain.
That they gala yako carpo carpuchin Teta Carolana and eco Lone de Carchilana, Lepig, Papa Gani, Luchuge, a lokichi, Shebuchi or what the and it you take out here to you, Lone Muxichin in the rest.
And you can see here that a person can be sitting and looking at a computer and doing a task while they're wearing an EEG cap, or they can just be doing a resting state while they're just kind of relaxing or even meditating, or as Ken Paller does a lot of research on people while they're sleeping.
De Carolina Sugar, let's go on the twisted carpet on China, and then do stop by Mana, meeting could saw that on the Lola, Loletisha, Loletic, Layanico, Cheguti, the Riach, or Tinder do dripping capsuco, Lepin on the Carcachi, do the Sajan decoration, I manage to Uncle Carsugata, Chilhulu, Depic Capsatan, Gomjapi Capsicle, let's Lepa Gungich, a lot in jet and a roach and decor anything on this, and I man again, Camp Holler. Somebody by now, that's a nippy cups, a young Chigalepagan on a Lodakana do tell a Tassi.
Now we can implement techniques to take this raw EEG data emitted from the brain and process it into kind of metrics of how active or deactive the brain is.
Chitantuchi or Tantu data Natu Turuchi de and a Tikalana quota, Iship Latin data, and you call and decapsuni, Lepate Chica Salon Chi, Tomporisha decapsa Salon Chi, Tompodusachorwa, or Tigitelaya, Padus Joyores.
And often people refer to how kind of active or deactive the brain is as power spectra.
Lepati Chica Salong Sete. Decision Stambalaya, Tela Karigon Sombrezana, Nick Topki, Tamses, power spectra, Top Tamses, Timu, on the decision.
So just to show you this a little bit more, when you look at a raw EEG signal, you get these kind of waves. They're basically waves of brain activity.
Ani Karsugureta, EEG li chombi, let's go on jelly chombi, Tachi Tantu, Tomiga Tantu Segura, in Se Machi Tonto Taba in Banachuga Gangi, Remodinach Tongris, the Decarelana, Chiga, Lepig and Anna Chombi, Lodag, Soyagi, and a lap in a race.
And we can convert these waves into these measures of power spectra to again get a window into how active or deactive the brain is.
That and they also Karcha and a Gala Chombi Tamputi, and also de Karsa top gig Tamsuganatele dos co Jura Latin data Latin date, and equal Lepate Karsugreta Salon Chain Domanduti dos Chayores.
And again, if you're getting confused, just think the simple kind of conclusion here is just that the goal is to basically get a sense of how active or deactive a person's brain is. And these power spectra just is just basically a summary of that.
ko selon katso chi sha selon chi yoba selon chi mepa gi tsete tongu yore swa tanda data ke ko chaya de kar song sena nga suka lepa te da selon chi ma je sena tongpo yo me sa ge ne ton song yore sti che su samna nang yo chis.
So EEG recordings are really good metrics of arousal or attention. Just again saying how active the brain is, how attentive the brain is, how engaged the brain is, etc.
다 담보텔라 가서 그 레스그 로제텔라 텐데 가리 하고래는 가서 셀롱 제베 네밥데다 아니 가서 그랬다 도는 가서 스테이트 그 요메게 네타 치고라티 참고 요레스타 데카스 임바나 지 가서 그랬다 도나 슈칠레 치트레트 그도 되나 신게 지 가서 그랬다 욕도레도 세대도 서거라 정보레도 되게 네탄데 참고레스.
EEG is also a really good technique for studying sleep states.
되나 신게 아니 가서 EEG 단데 레스그 로제데 가서 Because
it turns out that different stages of sleep have distinct profiles of EEG activity. We're very fortunate to have Ken Paller to be a part of the program because Ken is one of the world leading experts in studying the neuroscience of sleep using EEG. And Ken will give a whole presentation, I think tomorrow, on the neuroscience of sleep using EEG. And EEG is also really good for studying certain neurological conditions like seizure disorders and is sometimes used in the hospital. Now, again, different psychological or neurological states sometimes or often correspond to different profiles of EEG activity. And here you can see is a little cartoon of a person's EEG brain activity when they're really awake and really excited. And you can notice that the line is pretty dark and the spikes are pretty close together.
And
as a person gets more relaxed or sleepy, their brain waves will start to slow down.
And
here you can see the kind of the spikes get further and further apart from each other, or the waves get further and further apart from each other, and that's a reflection of the fact that the brain is slowing down.
지 타린디린드 드야도 지 통고도 왔다 되게 값이 제수가리 고그레스나 맞추게 레피 난게 지 제고시오와 대 지달래 두고 받성고요 레스.
And eventually, if a person starts to get really relaxed, they may start to fall asleep. And here you can start to see that the brain slows down even further, so that in deep sleep down here, you've got these kind of waves that are very spread apart.
다다데 가스 미치 니 컬러 차체 니트 스페셜 니트 조용 스탐발라야 코양 라트 아니 가스 그러다 베티 지타 조야다 네 다다데 가스가 니 지무 쿠비 값임바나 레피 난게 다치 제부 젤레 담데비가 라트 하창 도차요데스.
So again, we can kind of know whether somebody is awake or engaged or relaxed or sleepy based on their profiles of EEG activity.
진짜 다른 가수게 레지 로레기 그 파르치오로 파티 단다게 되게 랍통데 그 파르치도가 데이터라 테네데데 가수게 미치게 미티 아니 그 세데샤 엑사이드 차샤 양나 그 르르 차샤 양나 니트로데샤 양나 그 니쿠데샤 사이치거와 데이터 가수게 하굿 그레스.
So one measurement that I want you to be familiar with in EEG is called a hertz.
한다 현다 다른 가수 레스고 론제기 값을 네 지체제기 지시지 제출라 무트지 인세주 딩이 부레스 딩이 과데가레스나 hertz 디인스.
And it's a measurement of how many cycles of brain activity somebody has at that particular electrode per second.
나한테 되게 단다 hertz 디가스가라나 고다 론니스틱 값으로와 아니 미치기 레피기 나네 아니 고 karchachkanaatale ane koge jungkhor sagoro ata lok tanda labe mana lab rotan sagoro jungkhor te karchachkana thama katso chuchonsha sa te getsederes.
And to make it simple, basically a hertz is just a measure of how active the brain is.
Hanta tanda hertz sa de so de chi tab du zo ba ina karesana ko lepa te chi jongbo katso chiresha sa te getsenje ge chishires.
And higher numbers reflect typically higher brain activity, and lower numbers reflect lower brain activity.
Hanta hertz sa di chi tangka thoyo pa imbaina ani lepi ki che guti thoyo pa tangka mao yo pa imbaina lepi che guti ma chele ta mao yo pa chongores.
And you can separate these EEG signals into these different power spectra, which reflect the level of activity in the brain.
Hanta tanda ngasuge tioro karsa let's go lonjela tani chombe koge labchidwa lab data ani koge lab ki ta karsgore tangka ge tanda de koge chichong la tani de ani koge top ki ani chiga transfer se dinado yang koge mando la khatamya yores.
Now this is hard to look at, so I'm going to put in the chat a link to the spreadsheet that we have that goes through all of these terms, and you can study these in more detail if you wish.
ni chat box dina tangsha yes ani ke te chuga chi janye to ba na chela ya tus khachi chi ti kar karin da na digres.
And if you can't access it in the chat, this is on the Google spreadsheet for the course, so you could get it there.
Na ta tanda chat tangsha te la ya chi yang marong pa do bu chong ba na namjing su gen zen dra na la ya ani chu iksha tin da tamsa chi to ti na la ye ba res.
But I just want to make a few summary points of what you would find in that spreadsheet, just so you're prepared for the lecture that Ken will give, and also the tutorial we'll give you on the EEG systems on Saturday morning.
상이 괜찮라기 전달해야 되나 신경 채춘 전달하는 집이 값을 상이 수수수게 전달 속바 드라이치거로 대 값을 지그레스티 갈라나 지야 데이터 아니 전달 데이터 단 드라이브 비츠 네스키데 가시진스.
Now when a brain is very awake and excited or engaged or active, typically you see a fair amount of what's called beta power in the EEG signal.
Tanda ngasugi karsiga lepa this trombo yopa tan salon shuchambo theyopa ina chesuge tanda ge loji degnana bu degnana kari thongre sena ani ngasu EEG ge beta topse se de thongres.
And I realize this is a lot of text, but I just wanted to copy exactly what was in that spreadsheet, just so you see it, how it's in the spreadsheet.
Tanda de chugene tsuma bushto wa de tanda chetsula ji rimuchi tamsha yorwa tanda otegnana kari yopa de shaja dustrisha yes.
But all I want you to think about when you think of beta power is that the brain is active, and it's doing, this is often corresponds with doing conscious activity.
Tanda digi better to say de chitangi bana ngasu chiga samshin to dripping chegutin the dripping kapsa and better to say sending one grace.
Now, if you start to get a little sleepy, or maybe also a little relaxed, you have, and I appreciate, again, there's a lot of text here, but I just copied this exactly as it is. You start to have increased alpha power. So alpha power is something that associates more with kind of relaxed states of brain activity. The higher the alpha power, the lower the brain activity at that particular electrode.
Power sage or the photosynthesis to biokinetic capsa and a lonely titan David taking Damla Yuchuga Paten and get you Chegutin.
This may be a reflection of being relaxed, or it could mean that the person's getting sleepy.
The big Karisongu and I made a Toshicisa Disuguroko, a Yamana could relate how you patan Yesuteo Patayana Kupa, Nitu Patitsungures.
Then another kind of signal of EEG activity is a signal that we typically see when somebody's in a really deep state of sleep. And this is what's called delta power. And don't worry about memorizing all these terms. I just want you to be aware of the fact that delta power is, something that we would see when somebody's really in a deep state of sleep. So, for example, in some of Ken's work, he'll have people fall asleep with an EEG cap on or an EEG system on, and he can tell what stages of sleep they're in based on their brain activity. And what we would like to do over the next, few years is to start some projects with you all where we can study the neuroscience of sleep using the EEG systems that we'll send you. And these EEG systems can also be used to study certain emotional or cognitive states, as well as the effect of meditation on the brain. Now, this is a picture of the systems that we're sending you, which is a much simpler looking device. And this device has a few EEG electrodes right on the other side of this little band here that you can wear as essentially a headband, and you can also sleep with this on, and it will measure your brain activity while you're actually sleeping or while you're awake or doing whatever it is that you're doing. So here is a picture of some data that we got from the Muse system. This is called the Muse EEG system. Actually, just call it the EEG system. Muse might confuse. And you will be able to collect these data on yourself. So this is the picture of data from James Glazer's brain, who's my graduate student.
James
So let's unpack this, or let's explain this. So this scale here, you can see my cursor moving, Dawa?
Yes.
This scale here, this is what's called often the vertical axis or the y-axis in a figure. And I introduced to you earlier the term hertz, which just means how active the brain is. And each number here refers to how many cycles per second the brain is oscillating at. So James put this on before going to bed. And this section here, don't worry about this. This is just kind of motion artifact while he was putting it on and getting it prepared. Right about here is where things get interesting. So hot colors here, if something's really red, what that means is that there's a lot of that particular power spectra in James's brain. And as you can see here, James is getting kind of sleepy. He's having a lot of alpha power or alpha activity, which is associated with that kind of relaxed but sleepy state. So this is, you can see this band here of alpha power, which is associated with James getting sleepy and getting tired.
And it could top city carries and alpha dot cities. The day caps alpha dot setting as such Tele Chapatang, Niku to edge or I need to again. Netantan or something that they caps James La and a Kutele Chapata Niku batteries.
But before falling asleep, you can see James also has some brain activity across the entire power spectrum. So he's got some activity up here in the beta range. So his brain is still firing at, kind of high rates across the spectrum, but it's really the alpha power and sleepiness that's taking over.
그 랩빅이 나라 지금 가서 그랬다 lokki loda te tangya te shu chambu yo pa yang ko mambuchi dua du sro ko tamse te mambuchi du imba na de ka sa zo de da ji tele cha ba dang ni khuya gi chu ni khu ni tu ya gi ni che go te shu chambu yo pa ten de shas.
But right about here, James starts to fall asleep, and you can see that his the activity of James's brain is slowly kind of descending, and then at this point, James is now fully asleep.
Ta ta de ge pa ro de la ya ane ko ge le pe nang gi che go de jinyon chin che ta da ge sam de le stamba la ya ne be te ni le khu ba de chung go res.
And again, the red colors here, all it's saying is that power spectra is what's most prevalent or most present in James's brain.
Ta da de la ya marpu chi to de la marpu de ge kar song gar sana ta da de ge ne kap de la ya ani James ge le pe ge na la ya ani chi top se And
now what you're seeing is a lot of delta activity in James's brain while he's sleeping, because this is reflective of very slow activity, very low activity in the brain. So he went into delta, a deep sleep pretty fast.
And again, this
is a device that we'll soon be able to work with all of you, and we can study cognitive, emotional, and states of sleep.
That day, Tandaga Yuchish to a day, Chitsugan Jobo Pechetang Ragres, that day attended the Tanga to Ching Zing Tang, and Sam Kamtan, Tanda, and a Nigan Ebertelaya, Tosicina, to Pechetang.
And on Saturday morning, James will lead a workshop with you on how to use this system.
And the Pembaga Shopala and James Kidus and subject integrative cups in Banakoge. And
Ken will also discuss some instruments that he's using to measure sleep, which actually involves a ring that you wear on your finger that can also measure with pretty good precision your sleep rates and some physiology.
Your restaurant, tea, or Koyang restaurant, and a dig and you could pick ups and a chicarsugreta, Zupunguchula sopa, Shangula sopa, or Tetala Yatosicireta, Tayang.
Now, I'm only going to spend a few moments on these next two slides because I just want to introduce you to this term.
That under dig jela Yachetoya Karsa, Chitulaya Tsikashimotochians.
Using the same data we get from EEG, we can also analyze the data in a slightly different way to study what are called brain potentials, or what are sometimes referred to as event-related potentials. Now, you'll typically look at these event-related potentials, I'll call these ERPs, in response to specific stimuli that you present to your participant, like a picture or some task on a computer monitor. Now, where EEG is a measure that really studies how active or deactive the brain is, ERP techniques allow you to look at more precise psychological states in response to the specific stimuli you're exposing the participant to.
ta question tin de chita pa chila ya tembi kapse chisu samlu na la ya samkhantan drewe ge ko karsugreta niba panda no chire sha se ti ne tashi chigore te la tagi yores.
Don't worry about all the details here. The main point I want you to take away is that ERP allows you to probe more specific psychological states where EEG just measures the general level of activity.
Ta da de la yo ko ta jiwi kapse ko nyokta chawchi yore de la shu sanama ta mascho yo mas ta de kapse chisu mi gwati karla na EEG sa tigirwa ngasu lesig loje ti kala na chitang gita lepi ki chayong ne ko tongpo katsuri sa trong le katsuri du sa ti tsenje ji reswa ti che ne ta de la ye chongje gi nise sa di ya pi sa ti ki kar chongore sa na ani ji karsugureta chongwa ngichin chi kapsrwa ta chongwa ngichin chi kapsa ani ko ge lepi gi na ne ko ge nibati kanda do ji resha sa te sa yores.
So you now know a little bit about EEG and ERP, and I'll return to these terms that we introduced earlier called spatial and temporal resolution.
Hanta tanda di kapsu shetsu ge letsu lonje sayate tang chongde ge nitse sayate nche haku yezang ni tanda nga tsu ge yangchi tangpo deshe je sha ju tskin la yangchi chikta ge yins tskite tangpo di chongje ge secha tang nyo de dunje ge secha res.
Hello. Oops.
Yes.
Can you hear me? Oh,
Yes.
it says my internet connection's unstable. I'm sorry. So you're able to hear me, Dawa?
Yes.
Okay. Yeah. So maybe you finished the translation. I just probably froze in an awkward position.
Oh, I think yeah. The one that you have mentioned, the EEG and the EPR and the spatial resolution, temporal resolution, I think.
Yeah. So EEG and ERP have very good temporal resolution. They're very good at studying the timing of the brain.
Tanda de kapsunga sugo let's lonje se te tanda de tinje se cha se chorwa jisi jimas la karsa chondre gi nise se chorwa de nicha la ya kari yago yore se na ananga sugo tinje ge se temporal resolution tinje ge se pe yago yore.
But they don't have very good spatial resolution. They don't really allow us to look where in the brain the signal's coming from because we're studying the signal on the scalp.
That shenda ko kari chunyo resistant balaya chunje ge sechate yagoyo mariste nichala karchila baina koge karsogreta kati yungu patita haku yo resroate koge imbaina ya chitangi mate jesa te kuram gu papa gala karsa yoro gu papa gala karbi kasi imbaina kani yungu yo sete ko yagotent kamares.
And so techniques that complement EEG and ERP to look inside the brain are what we'll talk about next.
Tanda de kapsim bainer what tanda geta EEG ta ERP la ro tanda ga so ge let's go lonje data ani chondre ni se de nichala ya ani chikar so greta pentro chine ka kung ngu ga la ya lepi ge nang la ya ni ko la jont ke do se ti set ke ge ni ko tablen ta chi chi gore ti tanda de shi jagens.
So this is where I want to introduce you to a technique called MRI, magnetic resonance imaging. Now, whereas with EEG, you wear a little cap that measures electrical outputs on the scalp. This is an MRI a picture of an MRI machine right here.
Tanda ngasugi EEG kapsimre let's lonje kapsimba na karchugore chesuga lepi gang la gopak gang la ko loni yechitina do konde to koge lepi gang gisgo da lepi nange lokada ti chenje gi yorwa ni shenda de kala na tanda de kablan yende perlang gi shenda de resta de wala ya pashto de kablan yende perlang MRI address.
And what you do is you actually lie on this little bed right here, and you're pushed into the kind of middle portion of the MRI machine, and then it can measure the structure and function of your brain. Now, MRI is sometimes referred to as a neuroimaging technique because the objective is to actually create images of the brain as opposed to measure electrical potentials on the scalp. So this is a image of a brain using a structural MRI technique. This is the same brain. We're just looking at it from 3 different angles. This is actually the highest resolution MRI image of a brain that exists. After a person died, they donated their brain to science, and then they took the brain out and put it into an MRI machine and scanned it for 100 hours. And you can see what great detail you're able to see these beautiful brain cells with using this technique. Now, don't worry, we don't need to remove somebody's brain and have them die in order to do an MRI study with them. Rather, we can use an MRI on a living person. Now, the physics of MRI is pretty complicated, and I don't want to get too deep into the weeds on it. I just want to make a few general points. And here I just want to say that MRI scanners use the combination of magnetic fields and radio waves to generate images of the body, including the brain. Here is a former student in my lab, Ajay, and 2 postdocs of mine who were playing with the MRI machine. And so we slide Ajay into this big device here, the MRI machine, and then we can get a picture of his brain. And this just shows you that MRI has really good spatial resolution because we can get very nice images of the brain as compared to an X-ray, for example, which doesn't give us very good images.
MRI so the carbon the problem picture by bana spatial and chunji sechati hajan so yopati tont cresta delata by bana matsuga lokper tan carbon the problem pertinchal drupa in bana gandelaya lokpertilaya chi kogi chashi sawushirame baje o telaya so carbon the problem attended john we partilaya koge kasugata chokawala sajakarkari yume se tiga sechete hajan chambu yoris.
Now, just to give you a little bit of a teaser for how this works.
근데 가다로 지금 레카 지금 메디콜라 야네 대시 통통짝이스
MRI measures hydrogen molecules, and in particular, the protons of hydrogen molecules.
단다 칼렌인데 발란 되게 아니 그 가리 천재그래서나 아니 가서 그래 양도요 양도 하이드로젠 체제그래서 다 양도가 나네라 조대 바이다 양도가 나네 양도가 부두지요라 프로톤 지요라 어때 천재그래서
I should have said hydrogen elements, not molecules. So the hydrogen element. So, and just one more sentence on this. The reason why hydrogen is such a good signal is that it varies with the amount of water in tissue.
단다 데캅스와 마수고 양드티 펜 토포 요베게 가르사르티 요베게 데터 천재라야 펜 토포 요베게 준센치카레스 담발라야 아니 코 단다게 양드티가 망용티로와 고 차폰 폰트치킨 폰트고 나라야 양드 가르사 양드티 망로 요세야티 아니 폰트티가 나나 추망로 요발라텐데도 고 만도만도 요거와 챕터 요로와 티기탑 그레스.
And because different tissue types have different densities of water, that allows us to generate these really precise and beautiful images.
다때 조데가레스나 가지고 폰트브식 만도라야 고기 두세 만도요 빠지 두세 만도 스탄바 띵나라야 추고 두세 만도요 레스 틴다 인스탄발라야 아니 가지고 디지털 데 페이지 동안라 텐데티 아니 가서 파티 하정 세우틴데 야고틴데 자트요 레스.
And this just shows you some images using structural MRI from my laboratory, where we can actually look at specific structures that we care about in the study of emotion.
단다 데카심바나 단다 데라 퍼카시지요 바데 ngarange toshi khanane tanda ani jelegi akablende pelem pechu tande tan lepegi chashingi chingi chenla jeju partinda re ditala tene de tan lepi chashi te tan ne ku chele tan ne tanda chikar semnyong tan de pe toshi jiwa de lepi tan gores.
And in our conversation tomorrow on the neuroscience of emotion, you'll learn what each of these structures do in generating our experiences of emotion.
다 상ning zentana yang asukar chigresna lepi chashin data ke wa ngasu ge semyong ta ani chi nyamnyong tina do kari chi chengge yo message wa te to chisu jengres.
Now the last point I want to make today is I just want to introduce you to the term functional MRI or what's called Fmri, because it's such a commonly used term in human neuroimaging.
다 단다데라야 치치수 무트치 주야기 타마티카린 시스템발라야 젤리 단데비 칼렌데 발렌세 디 인스티카레스나 지 왕자기 텔라야 심지지위가 값스 아니 고 치와 진지지위 아니 고 측지 차비레스.
So a lot of the conversations that the Dalai Lama has with neuroscientists like Richie Davidson and others focus on studies that use Fmri or functional magnetic resonance imaging to study the brain.
다티 세고에 전선치카린스나 아니 군든 땅단다 다 체리바와 원서 체리바 다 리처드 데비슨 타부 값스 고 가몬 나야기 조지카레스 담발라야 아니 지 젤리 카브렌인데 발란 디페치 땅데 땅 아니 지 탁시 드립샤 주기 네토 망포강라 가몬한 게요레스.
Now, Fmri isn't better or worse than EEG. It just asks questions in different ways, and the two together can be really powerful investigations of the mind. Now, one of the limitations of the structural MRI technique that I just showed you is that it's a static picture of the brain. It's just looking at brain tissue in a very static or non-dynamic way. A benefit of functional MRI is that it actually allows you to look at brain activity, both at rest or in response to a specific task or image that a person's looking at.
Jelly coming in the present de la yako ginsangario races tambalaya take and lepaki chele de la kukarichi do search or what la tat cris tiancho get a natural chicken suvi capsi lepichelina degree and natural cheguchi dripping capsi lepichelina rola tat curious.
So fMRI provides a more dynamic picture of the brain functioning as opposed to just the tissue of the brain.
Tanda dege chili kablanya the palan degerwa chiga lepigi puntu taya maimba ani lepigi jele tan debigi ani chi party tent curious.
So this is a picture of a tour through the brain using fMRI.
Tanda dege parts to nin tintis to dekalana chili kablanya the palan ti pechu tan de tan lepig on like project utila which is.
And you'll notice, like in structural MRI, we still have an image of the actual brain structure here.
That day, Natsuge Karsana could Chako Chili Chako Tande Kalanya the Palanda Swata and the Degalaya Chako Tanting Grace.
But superimposed on that brain structure are these colors that you'll see.
Tatandagi Kogi Chakurwa, Lepagi Chako Partigal Tele, Kanaja Badeka races, Tambala Degalayako, Santandamrachtoa, what data is.
And the colors here are what the fMRI signal is telling us.
That dig on Gangla Yopi Santa Manda Dizu Karitongo Resena, any Jelly Kablanya Kalan Parlangi, Yanda Parlantegi, and Koga Karikarichi Songo Pati Songo Res.
And typically, hot colors like red indicate that the brain is more active in that region, whereas cool colors like blue indicate that it's less active.
Tsawatang Chawagi Santata in Bina, Dig Tsawata, Marpu, Santa Tikar Songoresana, Marpu Yosagalepi Chashi Telayako, Tikars Tupu Barwa, Trombu Batan, and each Tangwi Santa Tandawachi, Wombu Satelaya, Tigi Chashi Telea, Lepati, and Chelishi Mepa Yanko, this Reloyo Pasongores.
So this is the front of the brain here. This is the back of the brain. This is a person's eye socket, their nose, etc.
That is stop by Bana, Tanda Mapu, Tonsabu Yosade, Michi Lepagan Danguiche, and the Jandate Jaloche, and the Delaya, and Coparganana, Mitige, Mirigi, Miconteon.
And what this image here is showing is that the portion of the prefrontal cortex here, right above the eye socket, is very active or engaged during this particular task.
Tanda Tigi Karsongorasana, Tandagi Jegu Drippy Cups. Ade ngasugen dungunga lesson sigrwata lesson te ane ko trombu imbate songoris.
And so what we can infer here is we can infer that this portion of the brain is potentially really important for the psychological task that they're doing while they're in the fMRI scanner.
Tanda de kasu karichila ya jipado chitkresena koge ta jelegi kablanyanda pralangana la ya de kaps tasitripi kaps Nakola ya Jeguchi Dratesha Yorwa, and then Dickups, Jegu the Drip Cups of Kogita, and some canton picture Lichorwa, DNA kitchen boom but some yoris.
And this is a task in which a person is processing rewards, looking at rewards, and this portion of the kind of prefrontal cortex appears to be important in processing rewarding or pleasurable stimuli in the environment.
That other day Capsacarana, love the Toxe Chisachora, Koge. Lepig in Tangasun, Dungulation, Deganala, Chomba, Decarsongoresena, De Titandoyores, Nasugita, and Yepa Sigurta Chaga Sigura Chaga, Chagatan Joyu, watch a Chagachi Rago Pati, Corti, Samlunala, and Eco Yesechio Patitandoat in the rest.
Now we use the same tools to study fMRI as we use to study structural MRI. So we can look at Ajay's brain activity as well as his brain structure using the same machine.
That under the Kasanga to Trichi Chipa Pecheton, de Capsing Banagasuga, Casugre, Jelly Laya Tosicurity, Nansing, Asuga Chacola, and Tassicin Goris.
And I just want you to be aware of the fact that when you look at an fMRI image, typically what you'll see is you'll see the structural MRI image, but superimposed on that or on top of that are these heat map colors, which indicate more or less brain functional activity in that region.
단단데 가서 버르데라 따바이 반에 치수 통야지가리 통거래 세나가 데 이야다가 수기 젤리 갑len인데 발랭기 진샤지고 빠르치로와 데 값스 치수 통야데 아니 레피기 찾고 빠르치 통고 요레스와 찾고 빠르티기 간라야 아니 티 간라 가리 카는 치요레스 담발라야 레피기 니 차시 때 땅데비기 젤리 땅데비기 고게 particular tanda de la ji marpo sapuch to ting ku gala khana ji ore ti kar songors na lepe ki chashi de la ya ane nikap ta anja ku ge ani chegu kar drama de sa ji oro atti chong yores.
Now again, the physics of this are pretty complicated, so I just want to say something. I just want to summarize very briefly how Fmri works.
Ahanda tanda de ge jelly kablen de parlen de ge ko ning kham gi do ta te nyota cho yores ro imbai na tanda de la and
the summary is this: when brain cells are more active, they need more energy. The way that energy is carried to the active cells is through the blood.
정부 요백에 레픽이 차시 때라로 살롱 지위 레포이 차시 때라야 뉴바 간다스젠데 지그레스 스탐발라야 아주 쳐주지는 요레스.
And so Fmri indexes the activity of the brain by modeling where blood goes.
나한테 단다 고 젤리 칼렌데 발랭게 고그 지탕 주디 카레스 스탐발라야 아니 고 가사 레픽이 차시 관계 거라야니 참만도도세 텔라텐데 되는 거레스.
Now, if you're interested, the actual specific molecule that Fmri is sensitive to is a molecule called hemoglobin. Hemoglobin carries oxygen. And hemoglobin affects the magnetic field, and so that's how Fmri is able to determine where blood is flowing in the brain. And so you now know that the take-home points of Fmri is that it measures the brain activity through modeling where blood flow goes in the brain. And the last point I'll make here is that you can put somebody to an Fmri scanner and have them look at images in the Fmri scanner and model their brain in response to emotionally meaningful stimuli. Like faces are very emotionally meaningful. Snakes are spooky and scary, so we can study fear regions of the brain associated with that. We can have them do reward tasks, ways of provoking the brain in the Fmri scanner.
Jelly Kablan in the Parlangatina Chinu Jela Yan, Lepi Chashi, Conchite, Yamio, the Samuel Kangit and Rodu said, Tasachi Kasim, Bananga, Paris, or Hopper, the Dompa Parchi or what in Manangasu, Dompati Gatale, Namjan Dramdate or what they told Latin the Tanko, Namjan Dramdat Tamba Lepi Chashi Kanchis, Helon Chidu said, Tasachi Retina Shingi, a drug part in the Tamba, and a dream by Nangasu, thought the younger one. Otinashin tea time because saying Lepi Chashi Salon Chigu Kangila, Chili Dugu Chigu, that I already nashing Gaya, Shandanga Sugata, rewards like Doa, Nyambatra, and Nyambara Chigora, Nyambarata, and Chagara, Netanero Chipan, Titan David Kapsa, Lepi Chashi Kangi, Salon Chigu said, Titolaya, Musika, that Enco, Tito to be gone out of Sichet in the Yores.
Now, I'm aware I gave you a lot of information in this lecture.
다 단다 되게 람센데 나라야 쳐서 아니서 망보 지르셔야 yes.
And I also am aware that we didn't want to do a lot of lecturing in this research program.
단다 현대지 가래 하고 예 시스템을야 다시 심지게 sabjan데 가서 아니 지 가서 그래도 람센 셰 쇼로한테 망보 지 먹어야 돼야 하고 yes.
But I want you to be familiar with some of the terms in human neuroscience so that now as we move forward into some more experiential learning, you have a vocabulary to understand what these different techniques mean.
단다 데나나 이나양 단다기 과치라야 가속에 지 원서参기 다시나 로스타발라야 츠망보 시치 고속에 츠데달라야 지 무시야다 제마 츠데달라용 비캅스 지 카스브르다 카리실 고고도세데 아니 카스브르다 띵 니와지로 지스로데 갈라나다 나츠타 메트마 공부부바이마나 조데다 다시 심지게 지 제부드레치로 데간라 두그레스.
Now, you now know a little bit about the core techniques of cognitive neuroscience, EEG, structural MRI, and fMRI.
단다 디캅스 제수가리 하고 그래서는 인증 원서 생각이 나라야 아니 지금 배추당하게 답름 밖에 추어로 단다게 let's go 로르세레 테네 갑들서 착고게 젤리 칼럼 년도 발렌트 착고게 칼럼 년도 발렌트 되게 젤라야 젤리게 칼럼 년도 발렌트 사이즈로 왔다 지수 단다 하고 요레스.
And you may have some questions. So what I'd like to do is have you think of some questions and have you kind of think about these different techniques. And then at the beginning of my next talk on the neuroscience of emotion, we'll start off with a little bit of a conversation of these techniques. And you can ask questions. We can talk about the pros and cons. You can, ask if you think, whatever we want. We can kind of talk a little bit about them.
단다 데 제수가 데이터라 단다 탑람 가시지고 데이터 콜라야 아니 공파시 삼로다로 제스타 gelage 초탄 되게 지게 초탄 다다 3년 된 레벨 원서 리서치로 전자티 트비그 값스 전자티 고츠마 타타는 데이터라야 제수 공수 가르치예 데이터라야 아니 주게 당청 가르치도 공수 가르여 파타는 좋아진다 요 파이나 티 값스 양도스 ko lenja ta samsung at the chickens.
Okay, Nicole.
Great. So I'll just go over briefly what you will do tomorrow morning.
아니 그 도도지 선쇼바치도 가르치기 파티 시작이스.
So tomorrow morning you need to create or finish any surveys or interview questions that you need for your studies.
다 선쇼바치도 가르치고 그래서나 제쪽에 다시 심지디다 좋아요 주기 아니 전직이 좋아이나 되게 아니 제집 다툴어줘야 지고로 제집이 아니 차워지고 그래서.
And it is important to get feedback from other scientists, and so you will actually present your surveys or talk through your surveys with at least one other group before you finalize your surveys.
다때 양코 타마텔에 타미기 토초 마칭와라 야 체르파 신다게 사네 양코 공서 가르치도 세때 녀게 쳔보레스와 인스타발라야 아니 제추게 가사 제추게 땡캔 산노 제라야 아니 딘슨 신다게 사네 양 주 공서 렌지 나티 지로시스.
Once your survey are finished, if you're doing a descriptive study, you can start sending that link to people to collect data.
단다게 제쪽에 제집대 주바인바이나 게디 저런 게 지금 레지오게 제집들은 레지오게 prescriptive studies고 레지오 지역에 다시 되는 로직이요 바인바나 아마 다시 하는 게 제라 네 제수 신단 주시 진이라 두 다양 고치시는 리그레스.
If you are doing an experimental study, you should make sure that you know exactly who is doing what. And what your participants need to do or not do in order to collect data.
다 디마인 바탄다가 지금 레지오게 집제되는 마인 바지 다시게 집제되는 로치지기 요바인바네 제쪽에 니어다 하고 수그 가르치기 요메단 가르치지 지축고 요바단 가르치지 축고 매바게 네던데 서마 하고 요레스.
So for example, if you are for the coffee group, you need to decide before tomorrow evening, how you're going to have some of you have decaffeinated coffee and some caffeinated coffee, if that is possible.
Yeah, coffee tanda chu dinsan ti mai na ro ni sani chechu ta gonda gi jana ma chunga la ya ani coffee take yore coffee tong yore inda coffee ta susu gi coffee karsu gre ta coffee jindang coffee ti tongje yang kashi gi decaffeinated siguro ta ti coffee na ni caffeine jinwa tongji coffee chigoro wa oti tong yo ba ti kana ro chi chi ba ti ta che gu yores.
For the group that is focusing on watching a video of His Holiness, you need to know what that video is, when your participants should watch it, and which participants should watch it. And then if you have time tomorrow morning, you can also begin preparing your final presentation. So you'll create a presentation in your group folder, and you can start making slides about your question and hypothesis and study design.
Folder to what we just to attain on any so we presentation say they do. And take nalaya and she so we Karsugre Chesha Karimbe and Karsugre Tetala Nichi draw Karimbe, Drea Kanadochi Jibe, and the Tablan Kanadochi usage or Teta those another degrees.
And with that, we wish you good night, and as always, we hope you sleep well.
And as Janangos. You to pronounce